Transcription is a very interesting step in the central dogma. We've learned about DNA replication, and now let's take a look at transcription. So, DNA is like a coded book. We don't know what it means. DNA just codes for it, just gives us instructions. And the job of transcription is really to understand that code, to decode the message of that weird written language of the book of DNA. And by doing this, now we're gonna eventually make this very nice little transcript of messenger RNA, and it's going to say, okay, my DNA is saying this, this, and this, code for this, this, and this, and make protein X. And how do we make protein X? By the process of translation. But that's later. Let's talk about transcription now. Let's see how transcription works. So over here, I've already labeled some stuff over here. Here's transcription, and let's take a look at this picture over here first. Of course, my genome is very, very long. And if I want to transcribe a specific protein, if I want to transcribe a specific gene to make a specific protein, I'm going to take a specific part of the DNA. So let's just pretend that this part right here codes for protein Y. And protein Y is crucial for whatever, right? It's fundamental. It does something very important. That's not relevant, okay? All we have to understand, our goal here is to transcribe and then finally translate protein Y, so it can be in our body, it can be used in our body, and now we can function properly. Well, let's see, how does this work? Well, let's take a look over here. Transcription is a very fascinating process. Within transcription, we have something called a promoter. A promoter is located usually in the upstream direction, which is near the 5' region, and the promoter in this case is this right here. A promoter is a short segment of DNA that allows the enzyme RNA polymerase to bind onto. RNA polymerase, once again, is not so smart. Just like DNA polymerase, how it needs to recognize a primer, RNA polymerase needs to recognize a promoter site. And a promoter site also helps establish the fact of what you're going to transcribe. So this region right here will be our promoter. It's located in the upstream region. And this promoter will help RNA polymerase bind onto it. But now let's take a look at something. If I told you that I'm going to transcribe this way, this right here, this bottom strand right here is noted as my template strand. Well, what do I mean? If I'm going to make a transcript, by the way, transcription messenger RNA is single-stranded, right? RNA is single-stranded, so we're going to transcribe something like this, and I'm going to label this as a 5', 3' direction. Remember, if it's going this way, if I'm transcribing this way, 5' to 3', if this is my template strand, check it out, 3', 5'. It matches, once again. Always look for that pattern. So now we have transcription. I told you my gene is going to transcribe this way. It could, however, transcribe this way, but if it does, that means that this will be my template strand. So in this case, just for this case, this scenario, this is my template strand, this is my non-template strand. Let's see how we can produce this thing right here. So here's my promoter, and I told you RNA polymerase needs to come in. Actually, RNA polymerase is a pretty big enzyme. RNA polymerase is a very big enzyme, and we ultimately want to recruit it to the promoter region. So right now it's just floating around. By the way, transcription occurs in the nucleus, takes place in the nucleus, very important, very easy also to remember. And now, what do we want to do? Well, it's not so easy to recruit RNA polymerase. We actually have to have something called transcription factors, but we're going to talk about that in a second. You see this region right here, these TA. This is called the Tata box. We like to call the Tata box. And in this region right here, the Tata box, the RNA polymerase will bind onto. And to help RNA polymerase, to help facilitate this process, we need something called transcription factors. And these factors will bind on the promoter. These are various factors, proteins, which will basically help. So we have, transcription factors. One may look big, one may look small, so on and so on. And these guys will sort of help facilitate RNA polymerase to come in and bind to the promoter. It's going to shout, "Hey, hey RNA polymerase, come near us." So transcription factors basically facilitate and help transcription occur. We want to ultimately recruit RNA polymerase into this promoter region. But just transcription factors alone is not necessary. In fact, there's something called enhancers. And enhancers are a very important concept. Enhancers are things that allow to speed up the process of recruitment of RNA polymerase to this site. It doesn't necessarily speed up transcription, it speeds up of how fast RNA polymerase can bind on to the promoter. So once again, enhancers they help RNA polymerase to go in this promoter region. So now let's take a look at where enhancers may go. Enhancers are, unlike transcription factors where they have to be present on the promoter, enhancers can be present over here, they can be present over here, upstream, downstream. They can be located on intronic sequences, they can be located on exonic sequences. What is that? Exonic sequences and intronic sequences. Let's introduce that right now. Introns are non-coding regions of DNA. Non-coding and exons are coding regions of DNA. Or these code for something, introns do not code for something. Enhancers can be present on both of them. And now you might not know this, but most of our DNA is made up of junk, which is referred to as intronic sequences. It's not really junk DNA, but they used to use the term junk DNA, which refers to intronic sequences. They don't really code for anything. However, they play a very, very crucial and fundamental role in the central dogma of biology. But we won't get too much into that. But nonetheless, just be aware that enhancers can be present on introns or exons or upstream region in the promoter or even downstream. And these guys help facilitate RNA polymerase to bind onto the promoter. So this promoter region is obviously very important. Here I have RNA polymerase. It will finally bind. It's going to finally bind. It's going to unwind the double-stranded DNA. Unlike DNA replication where we have to have helicase topoisomerase, this guy is a one-man show. It does everything by itself. And what it's going to do is it's going to separate them and now transcribe a nice mRNA transcript. So we're going to go from here and we're going to synthesize this mRNA transcript. And of course, I'm going to label it 5' to 3'. It's a very messy diagram, so let's clean this up right now. But this is essentially how transcription works. You have a promoter region, the TATA box. You need transcription factors, you need enhancers, which will ultimately recruit RNA polymerase to the site of the TATA box, and now you can start to transcribe in whichever direction. So I mentioned this direction, therefore this is my template strand, and now what do I have? Well, what do I get now? Well, this is what I have. So from this 3' to 5' template strand, I've made an mRNA transcript. I'm going to signify this as a squiggly line, 5', 3'. This is my messenger RNA. But now we have to talk about RNA. We have to talk about transcription because RNA it's a bit different from DNA. So RNA, what are the characteristics of RNA? Well, it's usually single-stranded. You'll learn that RNA is always single-stranded. That's not really true. But for this case, let's keep it simple. RNA is single-stranded. RNA is the base thymine. The base thymine, which is T, is replaced by an uracil. And uracil is noted as an U. So let's try a practice problem right now, actually. If this right here coded for ATUA T, C, oops, U. This is DNA. You can't have U, that's not possible. T, A, G, G, A, T, A. Let's see. A pairs with T. This is my mRNA, but can I have T? The base diamine is replaced by uracil, so A actually pairs with U in this case. RNA base pairing rule. RNA base pairing rule. A pairs with U, U pairs with A, and C pairs with G. Okay, T pairs with what? Not U, T pairs with A. Technically, T pairs with A still. So T pairs with A, T pairs with A pairs with U, T pairs with A, C pairs with G, A pairs with U, C, C, U, A. C. This is going to be a U, A, and U. So here is our messenger RNA transcript. So here's our mRNA transcript, but this is once again oversimplified. This will be considered our pre-mRNA transcript, but our pre-mRNA does not have all coding regions. Our pre-mRNA, our RNA polymerase, which has now transcribed this nice little messenger RNA molecule, does not have all coding regions. It is not all exonic sequences. Actually, in fact, we have intronic sequences and exonic sequences. We have coding strands, we have coding regions and non-coding regions. So let's draw a very simple mRNA transcript, a pre-mRNA transcript. And by the way, we're talking about eukaryotic transcription. So here we have our pre-mRNA. And let's pretend, okay, so here I'm just going to make these boxes. Let me separate them a bit more. Okay, so let's pretend these boxes are all exonic sequences, and these portions in the middle right here are considered intronic sequences. So these are exons, these are introns. Coding, non-coding, coding, non-coding, coding, non-coding, coding, non-coding. Well, this might be a bit too simple. We can also draw it like this. You might see it in several ways. I can just draw a huge little box over here, and I can separate these, and I can just start shading and stuff. And the shaded regions are called my exons. These are called my introns. 5' to 3' pre-mRNA transcript. So obviously pre-mRNA contains exons and introns, coding regions and non-coding regions. And now something special has to happen. The first thing that happens is that this pre-mRNA transcript has to undergo RNA processing. This entire thing has to undergo RNA processing, a very important process. And this is, by the way, eukaryotic transcription, where the introns will be cleaved out. The introns will be removed from this pre-mRNA transcript. We do not want introns in our final mRNA transcript. Why? Remember, ribosomes are going to translate proteins by reading the mRNA strand, the messenger RNA strand. But the messenger RNA strand only reads codons. It reads 3 bases at a time, which are codons, which code for an amino acid, and those are only present on exons. So we would like to have a messenger RNA that only has exons. So let's see. RNA processing happens. We have spliceosomes, which will splice out these introns. So we have spliceosomes, which will splice out these introns. We have various proteins that do this. And what happens is, so let's get a little bit specific here. I will use the bottom diagram. So we're going to splice out the intronic sequence. We're going to splice out the intronic sequences. How does it do that? Well, it recognizes, these proteins recognize specific sites. you see the beginning of this intronic sequence right here? This is called the splice donor site. The end of this intronic sequence is called the splice acceptor site. And these various proteins and enzymes will recognize the splice donor site and the splice acceptor site, and it will excise out whatever's in the middle, which is the intron. And now we have an exon together. Same fashion, once again, splice donor must recognize the splice acceptor site, and we're going to remove these intronic regions. And now what do you get? You get a nice mature messenger RNA strand with an exonic sequence. So this is all coded. This is all coded region. This is an exon 5' to 3'. And now we have to add two modifications. We have to add a 5' GTP cap, and we also have to add a polyadenylation sequence. A poly-A tail. What is that? A poly-A tail. Well, this very long stretch of polyadenylation just means there's lots of A's in this sequence. So we have AU, A, and so on and so on and so on. This poly-A tail and this 5' GTP cap work together to stabilize this messenger RNA. It allows it prevents it from degradation and allows safe passage out of the nucleus into the cytoplasm. Because remember, ultimately this messenger RNA molecule must go to a ribosome. And where are ribosomes located? Not in the nucleus. Remember, transcription occurs in the nucleus, and translation occurs in the cytoplasm, where we have free floating around ribosomes that can recognize these messenger RNA molecules. So we have this mRNA. This is mature mRNA. It has undergone RNA processing. We have spliced out the intronic sequences, and now we have just the coding regions. And this will code, hopefully, for a protein, a functional protein. But let's clarify something real fast. So here, RNA processing is very important. RNA processing promotes variation. Once again, RNA processing promotes variation. You can have, so the same diagram that we just had, let's actually draw another one. Let's pretend here's my pre-mRNA, and here are my exon sequences. After RNA processing, I can either produce mRNA of this length, or this length, or this length. Keep in mind, RNA processing allows for different variations of transcripts. Why? Why? Because splicing is random. Splicing, it's not like we do it from here. Okay, let's splice out from here to here. Let's do it from left to right. It's not like that. RNA splicing occurs in a, well, we can say random manner, or we can even take a look at it the other way. Let's just say a splice donor site does not recognize this immediate splice acceptor site. What's going to happen? Well, the splice donor site is going to recognize the next splice. The splice donor will recognize the next splice acceptor site. So let's see what happens. So let's pretend, so here's my splice donor, my splice acceptor, splice donor, splice acceptor. If this donor guy doesn't recognize this acceptor site, what's it going to do? It's going to recognize the next splice acceptor site. So this entire region right here is going to be cleaved off. It's going to be removed. It's going to be spliced out. But hey, look, here's a small portion of an exon right here. Is that not important? Well, no. We have different variations of transcripts that can be formed. This is the important thing to realize. RNA processing allows for variation. RNA processing allows for variation. Keep that in mind. So, beyond that fact, we have two modifications, 5' GTP cap, a poly A tail, and now this guy is ultimately ready to be exported out of the nucleus and go into the cytoplasm, where it can be recognized. It can go and bind onto a ribosome, and we can allow for translation. So this is for transcription. We're going to talk about prokaryotic transcription in another video.